going to get started. I've just recorded. We are in week 11. This is our week where we are finishing up digestion and metabolism. And we have, what do we have? We have our test next week over the chapter 24 and 25 in lecture. Again, that test is considered your cheat sheet test that you are able to use your books and notes. But then when you get to the lab, you are expected to have closed book, closed notes, and the lab is over 24, 25, and chapter 23 and 22. So our lymphatic, our respiratory, our digestion. And one of your classmates made a good little review quiz that I posted. Hopefully everyone's taken advantage of the efforts of that classmate and able to do a good review on that. Again, you do need to make sure you are fully committing your ADI-LT labs. So that way everybody makes sure that when I check the ADI-LT labs, when I check on commit, everybody committed and finished it, that then I can give you the 5 points, okay? And don't forget, you do have those to do. Okay, so one of the big things about this chapter is it's about metabolism. And most of the time, when we think of metabolism from pop culture, from Scientific America, from the commercials on TV, we think metabolism is how do we lose weight? How do we lose fat mass and make our bodies, which Americans are, I think, what, 50 to 70% of us are overweight and obese. We are getting bigger and less healthy with that. And when we think, I want to increase metabolism, I want to lose weight and get back to a healthier BMI, a healthier body fat percentage. When in fact, metabolism from a chemistry, biochemistry, from a science point of view, is that you have a number of reactions taking place in your body, and those reactions both build molecules and break molecules. And so to have more metabolism would be that you have more chemical reactions occurring that are both building and breaking molecules. So when we think of babies and we think of children, babies, when they're born, you know, most babies weigh about 8, 9, 10 pounds. And then at their 1-year checkup, they're usually doubled, if not more, their body weight. So babies have a very high metabolism, and they have a high metabolism because they are building and breaking down molecules at a very high rate because they're growing and they're doubling their mass and they're doubling their size. Okay, children have high metabolism because, again, they're still growing. They're still building and breaking molecules down at a very high rate. They're still increasing their cell size, their cell numbers. Okay, when we get to this place where we consider growth stops, so somewhere in our 20s, our metabolism is kind of what is the amount of building and breaking and the number of energy that we need to expend and the number of calories we need a day to maintain our function. Okay, and so somewhat the basal metabolic rate of a human is the amount of energy, the amount of calories they need to run all of those ATP pumps. So remember, like back in the nervous system, we have a resting membrane potential. Sodium's leaking in, potassium's leaking out, and we have a pump that pushes out sodium and pulls back in potassium to counterbalance what's leaking in and out. Right? Well, that pump has to have energy. And so your basal metabolic rate is pretty much like, okay, I have to run these pumps to maintain basal metabolic rate, to maintain resting membrane potentials on all these cells. And then I'll have muscle cells. Right now, a few of my muscles in my bicep are contracting. So even though I'm not using my bicep, I have a few cells that are undergoing an electrical event. So sodium's coming in, and I'm eventually going to need to pump that sodium out. I'm letting calcium out and binding to troponin. I'm letting myosin hook in, cross bridge, and I'm letting muscles contract. And I need to replace that ATP. I need to pump the calcium, and I'm going to need to pump some sodium. That is going to be some calories that I have to, again, expend and get for just maintaining my muscle tone. So I have a little push on my lymphatics, a little push on my veins to help bring that blood back to my system, back to my cardiovascular system. So some of metabolism, again, has been, it's all chemical reactions, but we tend to harness and really focus on the chemical reactions that usually lead to energy be expended, glucose, fat being burned to make that energy, and then so we can expend it. And we forget that high metabolism is also associated with growth and with building and making molecules and making muscle cells and making cells able to grow. Okay, again, your muscle physiologist, they tend to study, again, what are the muscles by name, what are their actions, and then as you get into higher levels of, you know, I want to get a master's, you don't go get a master's in muscle physiology, you get a master's usually in energetics, which is how muscle tissue, and that's skeletal muscle tissue, cardiac muscle tissue, and smooth muscle tissue, how do those cells get their ATP to expend when they're contracting? How do they get that ATP made? What are their preferred fuels in differing conditions? Differing conditions would be at rest, at moderate to low intensity activity versus moderate to high intensity activity. Okay, and so that's how, again, a lot of your muscle physiologists go that route, and they become biochemists, and they become very much into how do I make ATP and what fuel source and why that fuel source, and onward and so on. Okay, and so people who eventually are considered muscle physiologists, their PhDs are sometimes in bioenergetics, in muscle energetics, in something that's, again, a little blend of physiology and biochemistry. Okay, and I bring this up because, again, while we talk about metabolism in the cells of our bodies, we're going to focus a lot on how do fuels broken down and generate ATP. But there is another side to the equation. If I want to make fats to build more steroid hormones, or I want to make more phospholipids so I can make a few more vesicles, or I can make some more endoplasmic reticulum, or I can make my cell get bigger and have more plasma membrane. All right, I need to make a few fats and make that into a phospholipid. So there is a part of, anytime I want my cells to increase in size, I need to build some molecules to make that happen, as much as I need to break down molecules to make energy be expended. Okay, and so when we look at, again, metabolism, reactions that are known as growth reactions are going to be anabolism. So again, anabolic steroids, the big thing, some of you might be too young, but like when baseball, it was legal to take andro. It was legal to take these testosterone precursors. So they weren't technically taking testosterone, which is illegal. They were taking precursor molecules, andro and androgen, to push skeletal muscle to build and make more actin, myosin, titin, troponin, tropomyosin, so they could build more sarcomeres. More sarcomeres meant more myofibrils, and their muscle cells hypertrophied and got bigger. And so again, skinny little 19-year-old boy became muscle man 25-year-old who was power hitting the ball at the ballpark. All right, those were anabolic pushes on the body. So they were trying to increase their metabolism, their body's rate of chemical reactions, towards more anabolic reactions to build more muscle, to build more proteins, to build more cells, to build more phospholipids, to make their cells bigger. Okay, and that was, again, a push they were getting a little help from through injections and sources of testosterone precursors. Right? This is what bodybuilders do. The ones that are like Arnold Schwarzenegger, he was in the Miss Universe, or Mr. Universe, I should say, pageant, bodybuilding competitions. Those are steroid legal bodybuilding competitions. And steroids are considered acceptable because in order to get the muscle to be as big as it's possible, they use steroids. There are other bodybuilding competitions that try to be steroid free, and it's become kind of this thing in the bodybuilding world where there are people that want to do it without steroids and people that want to do it and get as big as possible with steroids. So they just make their separate competitions. Who knows? We might get to that place in the Olympics or in other sports where everybody's doping, so we might as well just let it either be legal and we have races that allow it and then races that don't allow it. Who knows? Right? Now, when we think of public media and people who are always about like The Biggest Loser, they want to increase their metabolism to be able to break down more fat molecules, more glucose molecules, so they can expend and break down more of their body mass. Those would be more catabolic reactions. And again, for many people, our body will easily take input and try to build and get bigger. And whenever we start to try to chip away and bring that down, our body fights us on that. And so that's part of the reason why losing weight is so hard. And it's hard once you lose it to keep it off, because your body usually wants to build, wants to grow, wants to continue to get bigger and better. Not necessarily better, but get bigger. And it's hard for the system to then stop and slowly come back down and not then want to readjust back up to that thing. So that's why a lot of people who do a lot of crazy, they do gastric bypass, they lose a lot of weight, and then they don't change their eating habits, they don't change their lifestyle, and they'll gain all that weight back. Or they go on these, fad diets and they follow it strict for 3 or 4 months, and then, a year later, they've gained all the weight back. I know The Biggest Loser, for a little while, they were saying, like, where are they now? And again, some of the women, like 1 or 2 of them had babies, and they were like, I'm not weighing myself right now because I just had a baby. But other people, it was like, I know I gained all the weight back that I lost on the show, because once I got home, it didn't change that I wanted to eat pizza and barbecue, and I just reverted back. Because there was like one of the winners, he literally gained all the weight back that he had lost after he won the show. So even like the one chick that won, that Allie girl, she even talked about how she was like the role model, the spokesperson, and yet she started to slowly see the weight was coming back, and age, as we age, metabolism, the number of chemical reactions is going to slow down. So if you're depending upon, again, that higher metabolism rate of catabolic reactions to compensate a little bit for what you're doing and eating and your activity that you're not taking, it's going to catch up. Okay, so now let's switch gears. All right, so metabolism is about building and about breaking. Okay, we have 3 big macronutrient molecules we're going to focus on. There's actually 4, the 4th being nucleic acids that make DNA and RNA. But DNA and RNA are not normally going to be something that we stockpile to create us towards obesity or unload and create a situation where we're underweight. So they normally get spoken about in terms of our body changes, because again, nucleic acids are there, and we tend to have what we need to make and create new cells, okay? And protein, DNA to RNA kind of stuff, okay? All right, so the 3 macronutrients we are going to talk about that do fluctuate, we do potentially either not get enough of, we get too much of, and then we can manipulate them to be stored in a manner to, again, save for a rainy day that never comes, so we continue to store them and store them and store them in a form that then creates us to be obese individuals, are our proteins, which the building block of protein is the amino acid. And we have lipids, which the building block of the lipid is going to be our free fatty acid. And acetyl-CoA, which is a 2-carbon molecule, is the building block to make fatty acids. And then we have glucose, which is, again, our carbohydrate. And again, glucose can eventually be broken down to acetyl-CoA, a 2-carbon molecule that either goes into the Krebs cycle and continues down towards ATP, water, hydrogen, oxygen consumption, or too much acetyl-CoA, because we eat glucose and we eat breads all day, can be converted into fat and stored for rainy days. Okay, so those are our big 3 nutrient pools, and again, they come from carbohydrates, fats, and protein. So your digestive system works to get carbohydrates down to the lowest level being the monosaccharides, the single 6-carbon unit. So that's fructose, glucose, I think it's galactose, right? Your proteins are broken down to where they are individual amino acids. And so remember, the amino acids are like tyrosine, tryptophan, alanine, glutamate, so on and so forth. Okay, and then your fats, which are your triglycerides, they can be broken down into free fatty acids. Okay, and those free fatty acids then can be, through breakdown, broken into 2 and be used to make energy, or they can be reconstituted with glycerol. So 2 fatty acids and a phosphate make a phospholipid, or 3 fatty acids and glycerol make a triglyceride. Okay, and then they're stored in the fat, they're stored in muscle, they're stored in the liver, or they're utilized for energy. Okay, so again, we bring these foods in, our digestive system works through, again, the amylase from the mouth, the lipase from the mouth, then again we add the stomach acid, the pepsinogen, then we add intestinal lipase, intestinal breakdown with other enzymes, lipase, maltase, sucrase, and then from the pancreas we add pancreatic lipase, pancreatic amylase, we add chymotrypsinogen, trypsinogen. So we continue to add more and more enzymes that eventually get carbohydrates down to their lowest level, so glucose, fructose, galactose, fats down to their lowest level, those free fatty acids and glycerol, and then proteins down to their lowest level, which will be those individual amino acids. And we pull those molecules in, we send all that blood that's bringing all those molecules in to the liver, and the liver does checks. So this is part of the reason why your liver plays a huge role in digestion, because it influences, fat emulsification with bile by making bile. But then it also plays a big role in, okay, I'm on a low-fat, heart-healthy diet. Well, I still need fat if I want to grow some membranes, if I want to grow some vesicles, if I want to make some steroid hormones like estrogen and progesterone. So if my diet is so small in fat content that the liver sees I'm not bringing any fat in, the liver's going to need to make it. Okay, and I'm going to need to make fat into cholesterol and some fatty acids, so that way I can then create phospholipids, create triglycerides, create cholesterol to be able to then use fat in areas that I need it, like around my spinal cord and around my nerves where Schwann cells live and oligodendrocytes are to myelinate my neurons, right? Again, so the liver's a big place to see. I don't have it coming in by the diet, so I need to make it. Same thing if you're on Atkins. So if you are so low carbohydrate that your liver sees all the blood coming in from your food, from the stomach, the small intestines, the large intestines, and sees there's no glucose, and the brain, the neurons, only can use glucose to make ATP in order to function, then your liver is going to say, well, I have to make glucose, right? And I have to make glucose from whatever these sources are that I can make it from. So glycerol, some amino acids, and pyruvate if I have pyruvate available, okay? And so as the liver, again, is seeing what's coming in, it determines, do I build these molecules because I don't have them for the important places in the body that need them, okay? And then the other thing about amino acids is we're going to learn that some of our amino acids are what we call essential, meaning I have to get them from my diet because I do not have the ability to make them. So I can't take alanine and say, oh, alanine, I'm going to swirl and swirl these few little things and I'm going to make you glutamate. That's not going to work, all right? So there are certain places, certain amino acids that I have to get from my diet, because if I don't get them from my diet, I can't make them. And then there are other amino acids that I can, kind of make a little tweak here and there, and I can make them into a new amino acid. And those are the ones that I can, again, I need more alanine. Hey, I can make it from this, okay? Because I have a bunch of this, and I can make it from there, right? So that is what is going on with the digestive, with the liver. There's this like compiling of, here's what I'm eating, here's what I'm taking in, here's then how I can make the ATP with this cell population of neurons only getting their ATP from glucose being broken down. Muscle prefers glucose, glycogen, and fat depending upon the intensity of the activity. All my other tissues will use whatever is available based on hormones telling them glucose is available or not available, fat is available or not available. okay? And so again, there's a lot of interplay here on hormones, availability of nutrients, and liver telling cells, this is available, this is what I'm making, versus this is what's available and this is what I can't make, okay? All right, so when we look at, let's jump to, okay, question. If your blood sugar is high, why is it bad to work out? Okay, so actually, for diabetics, one of the things about glucose getting into cells, so this gets into insulin, okay? When glucose is high, insulin is supposed to be produced by your pancreas to trigger glucose uptake in muscle tissue and in some of the liver and some of the locations of the cells that can take a bunch of glucose in to keep the blood normalized. When someone doesn't make insulin, like a type 1 diabetic, there is a way, and it's not very well understood, defined, classified, figured out yet. But there is a way that exercise seems to get muscle, has the ability to take glucose from the bloodstream and pull it into their cells without an insulin receptor being involved. So there is a glucose transport molecule that brings blood glucose into muscle cells without insulin. So in some ways, some moderate intensity, some increased muscle activity for a type 1 diabetic when their glucose is hyperglycemic would actually be good to help get that excess glucose in the bloodstream into their muscles. So when I was pregnant with my daughter, I was a gestational diabetic. And so if I had like a piece of chocolate, or like if I made a gumbo and I ate a bunch of white rice, my blood sugars would shoot up, right? And I would go on a walk after my meals, and the walk was to try to get my muscle activity up, so that way I could get more muscle taking in glucose and using glucose at a higher rate, in addition to the insulin being released by my system to push glucose into cells, that I could then get my blood sugars to come down and back to normal at a normal average rate versus being diabetic where they wanted to stay elevated, okay? So Corey, I'm not exactly sure. Again, you're getting into a lot of the nuances specific to probably being a diabetic, and I don't think I have a good answer for you, because I would have to probably look it up a little bit more and do a little bit more research. Again, your situation being a diabetic, they don't understand all of it either. So there's some other aspects of the insulin receptor in a diabetic acts sometimes differently too than what it does. It does some different things, the second messenger systems in the cells. So we're getting into a little bit more than probably what we need to for this class. So we can talk offline, okay? Okay. All right, so let's talk again about, let's get to the glucose stuff. Let's get to the carbohydrates. All right, so again, this is kind of a structure for this chapter. For every one of the molecules of our three categories we're coming in, when a cell, and we're going to just call this a generic cell, okay? So this is just a generic cell here, all right? I can't see. This is why I wanted my new toy. It was supposed to be able to let me write better. Okay, this generic cell, all right, is going to, let me see if I can do that. So we are going to take in fatty acids. No, it doesn't work. All right, and those fatty acids, depending upon the hormones. So again, this is where, are we in a fed state or a fasted state? Are we in this post-absorptive state or absorptive state? So it goes back to insulin and glucagon from the pancreas, okay? So in some situations, again, where we tend to see I have readily available nutrients because I just ate and I'm in that 4 hours of just eating, many of these, again, building block molecules are going to be in high supply, because if I just ate and 4 hours after my meal, my small intestines is beginning to take in the fatty acids, take in the glucose, take in the amino acids from that meal and increase their amounts in the liver and in the blood supply, I am going to see that the cells are going to take in these nutrients, and they are going to be in high amounts and in excess. And insulin, because glucose should be in high amounts in the blood, is going to come up. And we are going to see that in most of these states, I have a situation where I have so much of these molecules that I can burn some of them and make energy from them, and I can store some of them for later, because there's so much of it that I can do, you know, lots of things. It's kind of like, you know, you go harvest strawberries, and I got strawberries coming out of my ears that I can make strawberry pie. I can freeze some. I can make strawberry smoothies, and I can do a bunch of things with it because they are in large, high amounts. Okay, and so when you think about that situation of I just ate, I have a bunch of glucose coming in, I have so much glucose that some of my areas, like my muscle, my liver, they are going to turn some of that glucose into glycogen. They're going to use some of that glucose for energy because there's just so much of it and it's readily available. Okay, and then when I get to that state of it's been 5 or 6 hours since my last meal, I am now in that fasted, non-absorptive, post-absorptive state. Now, I don't have a lot of glucose coming in because I haven't eaten any in a while, and what is in the gut is probably not going to still continue to put glucose into my bloodstream. And so that kind of situation is, well, now if glucose is not coming in, if I'm going to use glucose, I'm going to have to use it from my stored amounts, and that's my stored glycogen, which the liver and the skeletal muscle has available. Otherwise, if I'm a cell like in my ovaries or in my stomach, because I don't maybe store glucose and glycogen, I might have to then not use glucose and try to use fat and a protein and amino acid breakdown as my primary fuel making molecules for ATP, because I don't have the ability to use glucose because there's not a lot of glucose around, and glucagon is telling me I can't take any glucose from the bloodstream. Okay, so again, the situation of which of these molecules you'll use, which of these molecules you'll make, which of these molecules you'll store, all of that comes back to what is your hormone level, what is your last time you ate, absorptive, fasted, post-absorptive condition, and availability in some cases, and ability, like in the liver, can I make these molecules or not? Do I have the building blocks to do that? Bark? Park, P-A-R-K. Okay? All right. Now, one of the things to consider also is all of these molecules are going to get to this 2-carbon point. And this 2-carbon point I mentioned earlier is acetyl-CoA, right? And acetyl-CoA is the molecule that when I make 2, and I end up with 18, it can become a fatty acid, right? So whenever you hear about someone who, oh, I don't know why I have high body fat, I only eat carbohydrates. Well, if you eat 4,000 carbs a day because you only eat bread, that bread will, again, give you lots of glucose, and you'll store some of it as glycogen, but there's a point where you can't store any more glycogen, and the rest of it, you're going to break it down into these 2-carbon acetyl-CoAs. You're going to burn lots of it to make energy for your ATP, but you're going to have so much of these 2 carbons, you're going to have a bottleneck, you're going to have a backlog. And as you get a backlog and a bottleneck of these 2-carbon acetyl-CoAs, you're going to start to convert some of them into fatty acids. And that is how a person can eat only carbs in their diet and still have obesity and high body fat percentage, because the fat can come from excess carbohydrates in our diet. Okay? And the problem with getting this 2-carb is once you get to this level, there's no going back here. Okay? This step of becoming 3-carbon to 2-carbon is a one-way direction. So once the 3 carbons become 2 carbons, the only option to store excess 2 carbons is to make them into fats, right? 3 carbons can reverse, but not 2 carbons, right? So that is part of the reason why I can't take fat and make fat into glucose, because fats are broken down into 2 carbon molecules. 2 carbons cannot become 3, and 3 carbons are the only way to make glucose, because remember, glucose is 3 and 3 come together and make 6. Okay? So that's part of, again, some of the crazy little tweaky math of why I can eat a lot of carbs and still get fat, and I can eat a lot of fat and I can still be ketoacidotic. I can still have issues, especially with diabetes, where my body thinks I'm starving for glucose because all that I'm taking in cannot be converted and made into glucose for my brain to then utilize for energy. Okay? All right, so most of you have at some point in time heard about carbohydrates, all right? Carbohydrates are polysaccharides like glucagon and starches, okay? And they are usually, again, complex carbohydrates, so they are, again, a glucose is sometimes made as a hexagon, and when you start making hexagons bind to each other, 2 together is a disaccharide, right? And so 2 monosaccharides together make a disaccharide. This is sucrose, maltose, and one other I'm blanking on, lactose, okay? And again, when I put that third and I continue to build and make more, this is how I polysaccharide. And my polysaccharides are glycogen, which is what we make in mammals and animals, or it's starch, fiber, which is in some plants that we then can't break down completely and get into our bloodstream, okay? And remember, amylase, all right, its goal is to break polysaccharide into the 2 carbon molecules maltose, right? And then later on in your small intestines, you're going to have the small intestines and the pancreas contributing enzymes like maltase, sucrase, that can then break that bond between the 2 monosaccharides and form the glucose, fructose, and galactose individual monosaccharide molecules. Okay. Now, once we get to, here's the 6 carbon molecule. This 6 carbon molecule is what we tend to, again, bring into our cells. And under the influence of insulin, we push it into being burned completely down for energy, or if there's some excess, we allow some of it to be stored as glycogen. Again, that can be in muscle and liver cells. Most other places will not store it, all right? Or we don't take it in because either we don't respond to the insulin or glucagon is having its effect because we're fasted, we're post-absorptive, we're way past a meal, where glucose is, again, not coming in as fast as it's being taken and used by the tissues, okay? Once we get glucose into the system, most of you have heard that glucose breakdown, right? So a 6 carbon molecule broken down into 6 carbon dioxide molecules, all right? So the 6 carbons that are part of one molecule become 6 independent carbons, part of 6 individual molecules, okay? That process is a 3-step process, right? And it takes place, step 1 is glycolysis, where 6 carbons become 2 3-carbon molecules, right? That's in the cytoplasm of the cell. That usually gives us 2 ATP as a net gain. The rest of it is the 2 3 carbons, the pyruvate, go through a step and become a 2 carbon molecule, and that 2 carbon molecule joins the Krebs cycle and gives us some ATP, but gives us a lot of the molecules that are holding the energy that then enter the electron transport chain and generate the massive output of ATP. Okay, so just to break down again, glucose is the monosaccharide. Through the digestive tract, we take in disaccharides like sucrose, table sugar. We take in polysaccharides like glycogen and starches. And through the enzymes amylase and sucrase and maltase and lactase, we break those down into glucose, fructose, galactose. And it's the glucose and fructose that we then move in our bloodstream. and we bring in and we utilize when insulin and conditions are allowing us to make it into energy. Okay, and that breakdown process has 3 steps. The first step is our glycolysis. And again, I like this slide. You don't have to be able to tell me every step, and you don't have to be able to tell me all the intermediate stopping points, because that's biochemistry. But you start with a 6-carbon molecule, and step 1 is a phosphate is added, right? And so sometimes we hear that glycolysis actually takes energy to make energy. Well, where do I get that phosphate? I got it from an ATP. So enzymes that break ATP and liberate phosphates down are known as kinases. And so this enzyme is hexokinase. It takes a 6-carbon molecule and adds a phosphate, right? It makes glucose into glucose 6-phosphate. And this first step does a few things for us. Once glucose is now glucose 6-phosphate, it's technically not free glucose anymore. So it goes into a little pool in the cell of being a little bit separate. So if it's a muscle cell, more glucose wants to come in per the concentration gradient. Right? The second thing is this is the molecule that then can be under, again, the insulin glucagon hormone modulation turns into glycogen. Okay? All right. This molecule, again, has a phosphate. Ultimately, I got to get a phosphate on both sides, and then I got to split the molecule. Right? So the whole point of glycolysis is to get a 6-carbon split in half. Right? And in doing this, and again, it goes through the steps, you don't have to know them, I get 2 ATP. And I do all this without oxygen being required. All right? And so basically what I've done is I've taken the energy in this phosphate bond between carbon 3 and carbon 4, and I've used that energy to create 2 phosphate-phosphate bonds in 2 separate ADP, now ATP molecules. Right? And in red blood cells, remember, this is the only ATP they have when they need to pump out chloride or pump out bicarbonate. Okay? All right. Now, that pyruvate, again, is 3 carbons, has to become a 2-carbon. So it goes through another little step and becomes acetyl-CoA. This is where I can no longer, right, this 2-carbon, once I make acetyl-CoA, I can't go back to pyruvate. This is why I cannot make fat into glucose, okay? Because fat will always break down into a 2-carbon, and I can never make a 2-carbon back into pyruvate. All right? There's no way for me to add a 1-carbon, a carbon dioxide. I'm not I don't have the ability to do phosphosynthesis. Okay? Photosynthesis, word photosynthesis. Okay? So once I get a 2-carbon, and I add a 4 and a 2, I make a 6-carbon molecule, new 6-carbon molecule, citrate. Okay? And then 6 carbons, again, continues. I keep adding 2 because I'm going to blow off 2. In this process, I make 2 carbon dioxides. So 2 carbons are gone, so that's why I got to keep adding 2 to my 4 to keep making citrate. Okay? And again, I make a little ATP, but what I mostly make is molecules that are holding the energy in those bonds that originally come from carbon-carbon bonds. They're holding that energy so I can put that energy in phosphate-phosphate bonds. Okay? And then I get to my, again, oxidative phosphorylation and electron transport, and I'm taking those molecules holding my energy from the phosphate-phosphate bond, and I run hydrogens and electrons through the membrane proteins, creating a hydrogen-rich, this is a hydrogen-rich area, and I make a concentration gradient where hydrogen flows, electrons are absorbed, and I make a bunch of ATP. Okay, and that's a very simplistic view because we're trying not to get to biochemistry here. Right? So the net of what I want you to remember about glucose is 1 6-carbon molecule of glucose should net us somewhere around 36 ATP. Okay, 2 of those comes in the first part, glycolysis, where we don't need oxygen. The rest of it comes mostly through these oxygen-dependent means and mechanisms inside the mitochondria. Okay? So this is, if a cell is given a choice, the cell wants to eat sugar. The cell wants to use glucose to make ATP. Okay? And as long as insulin and glucose levels allow it, cells are going to make glucose into energy. Okay? And so all the cells in my body, if I just ate some, strawberries, some pink lemonade, with sugar in it, I am going to be in a happy place. All my cells are going to be getting the glucose they want, breaking that glucose down and converting that glucose into energy molecules and carbon dioxide waste products that then every time I exhale are being exhaled out. Right? And again, when I have the ability to make glucose into glycogen because there's so much of it, liver and skeletal muscle are going to also take some of that glucose and build it into glycogen molecules, a complex carbohydrate that can be stored for the 5, 6, 8 hour point when glucose is not readily available and I want to keep glucose available for my heart or for my cells to continue to use it even though it's not coming in from the diet. Okay. When I'm also in a situation where I am eating very, very low carb and or I'm not eating at all, and or I haven't eaten in a while and my glucose levels in my blood start to wane and I get to this place where I need to tell every cell that can use other fuel sources to use them so they don't use glucose, and my brain continues to use glucose because that's the only molecule it can use, I now need to come up with a way to keep adding glucose to my bloodstream. I'm going to have to make glucose. Right? So one of the things you're going to see when we talk about molecules being made or broken, if you see the word lysis, or that usually means I am going to break something down. Okay? When you see the word genesis, okay, that means I'm usually going to make or build something. Right? So when I'm in a situation where I need, because again, I have either fasted, I'm not taking in glucose, I'm at that point in my day where I haven't eaten in hours and hours and hours, I need to make glucose so my blood supply carries and maintains normal glucose levels so that the brain is constantly being fed the only fuel source it can use to make energy. I am going to need to make glucose. So I am going to undergo gluconeogenesis. So I am going to make glucose from scratch. Now, glucose is 6 carbons. So I need sources to make glucose that are 3 carbons. So lactic acid / pyruvate is a 3-carbon molecule. So if I put 2 of these together, I should have 6 carbons. Glycerol is a 3-carbon molecule. So again, if I put 2 glycerol together, I can have a 6-carbon. Some amino acids can be made into 3-carbon molecules. So some amino acids could potentially take 2 of them together, pull off the nitrogen part, and I can make 2 3-carbon molecules into a 6-carbon molecule. All right? So again, when I look at how do I make glucose, I have to look at alanine has potentially 3 carbons once I pull off the nitrogen group. Okay, when I pull that off, I have 3 carbons. That 3-carbon can quickly be made into pyruvate, and then pyruvate can work backwards to glucose. Glycerol is 3 carbons. Glycerol can be very quickly made into, again, an intermediate step in glycolysis that has 3 carbons. 2 3-carbon molecules come together, and I get glucose. Right? Lactate and pyruvate, again, it's all about whether or not I have oxygen available and I'm having a backload of pyruvate or not. So again, if I make lactate back into pyruvate, I can turn lactate back into glucose. One of the things about gluconeogenesis, this is only in our liver. Okay, and in very extreme situations, the kidneys may also do this. Why? The only place I have the enzymes that can turn these reactions that want to go this way into reactions that go this way, because they are only usually found in the liver, or in extreme cases, again, they can be found and made from the genetic DNA in the kidney cells. Okay, so your liver is going to be a source of potentially making glucose when you are in extreme need of it, because you are super fasted, super decreased Atkins, super dieting, no glucose coming in, and your body has to have a situation where glucose is always available for your neurons to use, so your brain and spine and nerves function normally. Okay, again, anytime I see genesis, I'm building. When I have excess glucose, so usually when I'm in a fed, glucose-rich state, insulin is going to push glucose into my cells. In my liver and in my skeletal muscle, I can take some of that glucose and turn it into the stored glucose form, glycogen, and save it for later when the glucose is not as readily available. And then when the glucose is not readily available, the liver and the muscle can take that glycogen, break it down through glycolysis, glycogenolysis, and that glucose now is liberated to be either used by the cells themselves, like in the skeletal muscle, continue to, in my fasted post-absorptive state, continue to run and make ATP for contractile muscles to work. And in the liver's case, it liberates that glucose and puts it into the blood supply to keep the blood supply at its normal glucose level. So typically when the liver is getting into that fasted, post-absorptive 5-6 hour point from a meal, it's going to take its glycogen and burn through glycogen first before it starts making glucose. And then after it's burned through all the glycogen and it doesn't have any more source of glycogen, then it starts really having to use gluconeogenesis to make glucose. And if the liver can't even keep up with the glucose making from gluconeogenesis, again, I said in extreme crazy situations, the kidneys will then start to kick in and help. Okay, so glycogen, when you think about marathon runners, again, some of you maybe have never considered running a race of 26 miles, but one of the things you'll learn is your body, you train for 16 weeks running the distance one for the pounding of the mileage, but you also train your body to better handle the stored glucose and glycogen stores to make the mileage and have the energy to make ATP for the mileage. And when you run a marathon, there's always this point where people talk about, especially a first or second marathon, where the bear jumps on your back. And for me, it was at mile 22. Mile 22, I went from running 9-minute miles, 9.5-minute miles, so I could only walk 14-minute mile pace. I mean, there was no thought of running. And that usually is defined as the bear jumped on my back. And my muscles, even though I wanted them to continue to contract and work at this 9.5-9-minute mile pace, I couldn't. And it was because I ran out of glycogen. And at that point, the only energy my cells could make was fat and protein, amino acid, to continue to make ATP. And the rate of those making ATP, the fuel into ATP, was such that I was walking 14-minute mile pace. There was no ability to go faster because there was no more glucose or glycogen to burn and get ATP for glycolysis to kind of keep things moving along. Yeah, that wasn't happening. And so, yeah, my 5th marathon, I was able to run the entire thing. I didn't ever get the bear jumped on my back, and it was because I think by then, through the training, through the years, and through the discipline, I was better able to keep my glycogen and glucose available through the entire event. Still never made Boston qualifying time, but oh well. Okay, so that's carbohydrate summary, all right? When, again, glucose comes in certain conditions, under certain hormones, I will push to save some and use some. When I get to that place that glucose is no longer coming in, I will burn and break down, again, in the liver and skeletal muscle to keep my muscles working. In the liver, I'll burn and break down to push glucose into the blood so it can continue to feed the brain. Once the glycogen is gone, then I have to only be able to make glucose, and I make glucose again from three carbon sources. I cannot make it from 2 carbon sources. That's why fat will never be able to be a glucose making molecule. Okay? All right, moving on to fat. Again, fats are these long chain carbons. When I break them down, I break them into 2 carbon molecules. All right. Again, glycerol comes from the break of glucose because I take one of those 3 carbons and I make this backbone molecule. That is why 2 glycerols can become glucose, not the fatty acids. Okay. When I break down fats, again, lipolysis. Lipolysis is going to be in cells. All right. It's a step of first, I got to take the 3 fatty acids and glycerol and separate them. Okay, that's step 1. The glycerol, like I said, in certain places, in certain locations like the liver, can be converted to pyruvate and can become, through gluconeogenesis, made into glucose, or glycerol can enter in and become a molecule for part of glycolysis to make pyruvate and then continue on to break down like a normal glucose molecule would. Okay, the fatty acids. Right? I do the breakaway of glycerol fatty acids in the cytoplasm. That's the first step of lipolysis. Once I get my fatty acids, now I bring them into the mitochondria. And in the mitochondria, I start to break off my 2 carbons, right? And that process, the second part, is then going to give me a 2 carbon molecule that acetyl-CoA can go into the Krebs cycle, join with the 4 carbon molecule and become citrate, right? Because again, an 18 carbon is going to give me 9 acetyl-CoAs, and I go through the Krebs cycle and I go through electron transport 9 times versus this only does it 2 times. That's why fatty acids give me so much energy versus a carbohydrate. Now, again, when I want to make fats, all right, I have to take my 2 carbon molecule, my acetyl-CoA, and in the liver and in adipose tissue, I take those 2 carbon molecules and I build them into something that's 18, 20, 22 carbons, and those can become fatty acids. Okay, and then once I get 3 fatty acids and a glycerol, I can make them into triglycerides. Or I get 2 fatty acids and a phosphate, and I can make them into phospholipids. If I make, again, a fatty acid and I tweak it, I can get an echinosoid like prostaglandin, which then I can use as a signaling molecule. Okay, so that is how carbohydrates and 2 carbon molecules can be made from carbohydrates, can turn into fat. And it's how, again, fats can be made, and fats can be, again, utilized by mitochondria and made into energy. Okay, and again, acetyl-CoA, if I put about 30 of them together and I make some rings, I can make cholesterol, and from cholesterol I can make steroid hormones, and I can make, again, lipid rafts in what do you call it? In cell membranes. Okay, again, fats are made with single bonds and double bonds between the carbons. Some of the places where the double bond can come, like what we call omega-3 fats that come from fish, we cannot make a double bond on our 3 carbon in a fat. Only fish have the enzyme to do that. That is why it's called an omega-3 fatty acid. And it's a fat that we cannot make in our bodies. So that's why we eat fish sources to get those kind of fats. Okay. Again, there are other essential fatty acids that we have to get from our diet because, again, we don't have the ability to make the double bond in a specific location. I think omega-6s is another example, right? One of the things about fats is if there's single bonds in every location of the fat, it's known as a soluble fat because it looks like, instead of like there's these little breaks and chains, these double bonds would mean there's hydrogens there, and it would look like a saturated molecule. Okay, and so saturated fats, again, are not going to be soluble in water, and we normally think of saturated fats as potentially fats that are bad fats. They're just fats that, again, are harder to deal with in our system, and it makes it maybe more unhealthy for our heart and for some of our organs, right? So many of our fats, because they're long molecules, they are not water happy anyway. So they have to be carried by a protein. And remember, the carrier protein is the molecule made in the liver known as very low density lipoprotein, low density lipoprotein, or high density lipoprotein. And those proteins help make a little pod that the fat can kind of be in, but the molecule can be suspended in water, right? And remember that these are, again, not bad or good molecules. They just represent, if I have a lot of fat in my body to carry, I'm probably going to carry a lot more of my fat with these molecules in my blood. And that's part of the reason why I would have high cholesterol. I have high amounts of these molecules because I have high fat being carried in my blood. Whereas low cholesterol, that could be because, again, I don't have a lot of fat to carry. And when I do carry, I carry them more with this molecule, the HDL proteins, and a smaller little density product. And so it's not as likely to be picked up and deposited in our membrane, behind our epithelial cells of our cardiovascular system when they are getting damaged by high stress and high friction. Okay, so again, none of these proteins, none of these molecules are bad, and we make them in the liver as need demands. So part of having a low-fat diet is if I don't have a lot of fat to carry, I don't have to make a lot of these products. I don't have to make a lot of LDLs, HDLs, and I don't have a lot of fat in my system being carried around by these lipoproteins. If I don't have a lot of these LDL protein carrying little molecules in my bloodstream, when my epithelial cells are getting stressed, they're not going to pick up and deposit fat and start the process of atherosclerosis to occur in my vessel. Okay? So again, lipids, I make them and I use them when I need them. If I'm not getting enough lipid in my diet, I will try to make lipid. Again, I can't make certain lipids like omega-3 fatty acids, so I do want some fat in my diet, even if I am completely low-fat or heart-healthy food eating. I want to try to eat some sources of fat like fish that can give me the good fats that I can then utilize in efficient and effective ways in my lipid rafts of my membranes, in my vessel walls, and in my different places, in my membranes of my cells, in my vesicle walls of my vesicles that then dock better in my releasing neurotransmitter or releasing hormone. Okay? All right, and the last thing, again, is protein. And again, proteins come from amino acids. One of the things I want to talk about with protein is there's always this amine group. And so anytime I'm going to make protein into a molecule for energy, I am going to have to do something with this amine. So my choices are I can take the amine and put it on another molecule, and then what I'm left with when it's gone is something useful that I can then use to make energy or use to make hormones or use to do something else with. And that just basically, I'm taking the amine and I'm like taking it from my right hand and putting it in my left hand, and I'm using my right hand. Right? So I'm not getting rid of it, I'm just moving it to another location. The other option for the amine is I break it off and then I have to put it in a molecule that I can get rid of it. And that's where I make urea, and I get rid of it in my urine or in my sweat or in some other locations to be able to, again, get rid of the nitrogenous waste products. All right. Again, protein breakdown and breakdown of the amino acids to carbon molecules that I use for energy is going to create these nitrogenous waste products that I have to get rid of. All right. The other place I get a little bit of nitrogenous waste is when I start breaking down DNA, RNA, and nucleic acids because they have those nitrogenous components as well. Okay. But for the most part, if I can keep amino acids and not use them, that's desirable because amino acids help me, again, make and break proteins all the time and enzymes. So I want to potentially not use proteins as my primary source for making ATP. Okay, so again, any reaction where I move the nitrogen is a transamination. Any reaction where I basically break the nitrogen off is a deamination. Okay, we don't have a lot of time and I'm already over. again, this fate of if insulin's running things or glucagon running things, if you're in a fed state versus a fasted state. Again, your book called them absorptive versus post-absorptive states. And again, if you look at what's going on, whether you're burning and using a fuel source, storing a fuel source, that's where it's influenced. And this is all hormonal. All right. The big thing is in a post-absorptive state, I am going to, again, not have glucose coming in, but I need to keep glucose available for my brain cells, for my nerve cells. That glucose is going to first and foremost come from any stored glucose, so my glycogen first. I am not going to immediately start making glucose unless I burn through all my glycogen, right? And so that's why people who go on diets, the first week of your diet, you are not probably burning fat yet. You are not trying to use fat yet for energy, because you are going to burn through all of your glycogen first, and glycogen, being a carbohydrate, when we store it, keeps water with it. And so that's why when people, if you watch shows like The Biggest Loser, they'll lose 30 pounds or 10 pounds or whatever, a lot of weight in the first week, and a lot of times that is the water weight. Where is that water coming from? That was the water that the cells of our muscle and liver were holding with it to store glycogen. And so they burn through their glycogen to keep their glucose levels normal. And as they burn through their glycogen, they no longer needed all this water, and the water was dumped. And so their weight loss that first week is just basically not changes in fat mass yet, it's changes in glucose being stored as glycogen and the water with the glucose storage being lost and let go. And then normally when week 2 hits of a diet, the weight loss is like 1 to 2 pounds. And again, that's more reflective of if you're losing fat mass because you're burning 500 calories a day, and then 5 times 7 is 35,000 calories of fat, which is 1 pound of fat. That is more reflective of actually losing fat mass, not water and glycogen. Okay. All right. You hear about ketoacidosis, you hear about ketones, that type of breakdown and use of fatty acids to create, again, energy. And again, fats cannot become glucose. I'm trying to make something brain tissue can use for energy when there's no glucose available. I can end up making these ketone bodies, and I create these waste products. And these are the waste products that make people who are true to ketoacidosis have their smelly breath, right? That smelly ketoacid breath. It's because they're exhaling out acetone, acetoacetate, and beta hydroxybutyrate. Okay? And again, these are acids, they can accumulate, and the keto bodies and the keto acids are being made, and you start to create a situation where your body becomes really acidic really fast. For diabetics, that is very dangerous because they can get this way within 24 to 48 hours of just being sick, and their body not making glucose into, the system, even though glucose is there, and their body's starting to make massive amounts of keto acids quickly to try to, again, it's starving, make something for the brain to use as energy, right? And ketoacidosis is a big metabolic issue that can lead to death. So those of you that want to work in and around emergency rooms and deal with people who are obese and diabetic and they're not controlling their diabetes, you might see a lot of metabolic acidosis walk in. All right. Last thing is, again, the minerals. Know these for your test and have a little of this note taking near you, and then know your vitamins. Again, know what your vitamins are, which are fat soluble, which are water soluble. Those would be nice things to be able to ask matching or multiple choice questions on. All right, we don't have a lot of time. I'm going to stop the recording. Are there any questions for those of you that have stuck with me the whole hour?